So I'd like to turn now to the lecture tonight. In addition to our lecturer, Dr. Verena Tunnicliffe, we will also have two respondents after the lecture has taken place, and then we'll also have time for questions. I'll just give a brief biography of each of our major participants tonight. Dr. John Nightingale from Vancouver has a broad professional background in aquatic sciences, using that background to enhance public awareness, education, and to promote conservation into all things marine. Dr. Nightingale has been responsible for the development and operation of some of the world's best aquariums, and he's been a leader in the rapid development of new conservation efforts in aquaria, zoos, and museums. During his 17 years in Vancouver at the aquarium, he's guided the expansion of the aquarium's leadership in conservation and research while focusing operations on sustainability and solid fiscal performance. As a result, the Vancouver Aquarium is one of the, quotes, greenest cultural institutions in Canada, and it's the only one that's financially self-sufficient, operating without government subsidies. Dr. Paul Snellgrove is a professor at Memorial University of Newfoundland in the Ocean Sciences Centre and the Biology Department. He holds a Canada Research Chair in Boreal and Cold Ocean Systems. His research focuses on larval transport in marine environments, as well as the factors that contribute to natural patterns of biodiversity and their link to delivery of key ecosystem services. Different projects in his research area have focused on sedimentary habitats from near shore to deep sea, from invertebrates to fishes. He chaired the synthesis group of the Census of Marine Life. I think this is a project that many of you will have heard about recently. And that project oversaw the final phase of the program that has been, I think, now quite well publicized, and some of you may have seen the book that came out on this recently. He's now director of the NSERC Canadian Healthy Oceans Network, a research collaboration of 65 marine scientists from coast to coast in Canada. And obviously this project is continuing the work that the census initiated. And leaving the best till last, I come to Dr. Verena Tunnicliffe, who is our speaker tonight. She is the director of the Venus Project, which stands for Victoria Experimental Network Under the Sea, the Subsea Observatory, and she is also Professor and Canada Research Chair in Deep Sea Oceans at the University of Victoria. Her research focuses on the ecology and evolution of hot vent animals and marine community history. She collaborates widely across many disciplines, from geophysics to molecular biology, and I think she's quite well known for her work, her field work, which often involves the use of submersibles and some really stunning images that so many of us have seen. Dr. Tunnicliffe is a member of the Scientific Advisory Committee of the Canadian Healthy Oceans Network and the Governing Council of the Northwest Association of Networked Ocean Observing Systems, which is part of the National Oceanic and Atmospheric Association. I have to say on a personal note that I've certainly followed your work for many years, and I find it absolutely fascinating, and I'm really looking forward to hear you talk tonight. Dr. Tunnicliffe will repeat this lecture, I believe, in a number of locations across Canada over the course of the next year. So we're very pleased to welcome her tonight to Simon Fraser University. Thank you.
Thank you very much, Dr. Driver, and thank you all for coming out tonight. It's sometimes kind of hard to be a marine biologist and an oceanographer. When you step out into the oceans and start looking, we've got a lot of problems, and sometimes the profession is looked at as one of the more depressing professions to take up. But I'm not going to go there tonight. I want to share with you tonight the things that make me feel delighted to be able to go out in the ocean, the things that I feel should lift up the soul and inspire us to think more about what it is we're doing with the oceans and how we can do better. So what I'd like to be able to do is talk about deep ocean exploration, and most of what I'm going to talk about is somewhat related to research that's gone on in my lab or with my colleagues, and most of it will focus on the very deep ocean, and part of the theme going through it is how new technology developments have taken us forward to be able to explore in more detail or do things a little bit differently. This is a photograph of our beloved Strait of Georgia, looking down from above. And what we see is the sun reflecting off a surface, which is part of what our problem is. Light reflects off the surface of the ocean. We can't see in it. The human species is a very visual animal, and we like to be able to look at things. So out of sight, out of mind, we don't know what's going on inside, so we don't tend to pay attention to it. It's a very difficult world for us to get into. But there's always been that desire to get in, and starting in the early part of last century, such as William Beebe here in the bathyscaphe that went down to 1,000 meters depth, suspended on a cable from a ship, first entry into the deep ocean, and that taken forward by people like Jacques-Yves Cousteau, who was dedicated to filming to be able to share with the public what was under the oceans. And I grew up as a child looking at Cousteau's books and movies. This particular vessel, he was able to fly free of that cable and to be able to use thrusters to move around. So what we want to do today is get down through the surface, but be able to get down quite deep. And I'm going to take you on a little trip that's going to start out here in British Columbia, and then we're going to move over to the Mariana Arc, and then over to the Celebes Sea, to Indonesia, and we're going to do round trip and end up back in British Columbia. Are you okay sitting there, or would you like to move? You right? Okay, good. Now, to do these expeditions, we need a vessel to get out there. And over the years, we've used many different vessels from many nations, such as this one. This is operated out of University of Washington. The Thomas G. Thompson, typical big A-frame on the back to be able to lift our submersibles over into the ocean, and it's a global class vessel, so it can travel a long way. And on board our ship, we're going to take a remotely operated vehicle. We've done a fair bit of work in manned submersibles, and I'll tell you there's nothing like being on the bottom of the ocean yourself. Wonderful, but to be honest, there's some limitations in that. There's just two of you down there, and you cannot share it. The thing that I love about remotely operated vehicles is that there's a whole group in that control van. You've got people from all disciplines all trying to work out what's going on, and you're doing that development as a team. We've worked with a number of different ROVs over the year. Jason from Woods Hole. I'll show you some imagery from this little guy here, which is basically a gorgeous flying eyeball. And Jason and Ropos, which is a vehicle stationed over on the island, but it was built here in Burnaby by ISE. Both of these submersibles are real workhorses, and they have manipulators on them. And over the years, we've developed Ropos from being not terribly capable to being one of the best in the world. So for the first dive, we're going to launch Ropos, and we're going to start off a little bit shallow, and we're going to go down to the seafloor. So off we go. So I just want you to remember that it is dark down below 200 meters. And so that all the images that you're going to see are taken with lights. So to be able to see, we've got to take our lights with us. And exploring the seafloor is a little bit like going through the Rocky Mountains with a flashlight and trying to figure out and put it all together and figure out what the whole seafloor looks like. So let's turn some lights on. And this particular image is taken at 2,000 meters, about 400 kilometers offshore. And I have to say, just about everywhere I've been on the seafloor, there is the mark of mankind there somewhere. This particular piece of paper was a memo from the U.S. The Postal Service, here you can see, the U.S. Ministry of the Environment. We sent a picture back to the person who sent the memo. So the first place we're going to start is at the top end of British Columbia, right on the Alaska border up here. And we're going up here with the Coast Guard ship Tully. And this is part of a program working with DFO And as a matter of fact, I'm going to talk about a little bit of research being conducted by Charisse Dupree, who's sitting over here. And she's doing work with the remotely operated vehicle to develop techniques for quantitative mapping, and how is it we can do, instead of just gee whiz pictures, how can we really create replicable science and quantitative methods on the seafloor. And then the particular question is addressing fish and corals and sponge relationships. So if we look at a map of Learmonth Bank here, it's about 150 meters down on the surface, and there's a big seafloor around here, which is a great fishing ground, some of the best fishing in British Columbia. And the area around the southern area of the bank is quite heavily trawled, not trawled in the northern part. So with this kind of imagery, really good imagery from the submersible, HD cameras have only just become available in the last couple of years. And most of my images, you will see a couple of dots on them, laser dots. Most of the scales are 10 centimeters apart. And this particular study is looking at the bumpiness of the seafloor and how the animals respond to that bumpiness. Here, this image, we've probably got a coral that's been broken by a trawl net, and certainly evidence here, a trawl net caught up on the bottom. The corals in our waters are absolutely spectacular. This is the red tree coral. And usually when you see them, they can grow up to about 2 meters in height, and they usually have somebody associated with them. There's a couple of fish down here, a little octopus, a little crab. So here we are going across the bottom, a couple of thorny head fish here, and we're looking at the bottom. You'll see boulders in which the boulders create a habitat for animals, and looking at how the roughness then creates a surface for animals to settle on. That's a hagfish. The rockfish are often taken along by the currents. They like to, they have to spend energy to keep in one place. This is a big mouth sculpin with a beautiful face on him. And here we're coming into a coral that's sitting on a large boulder, and you can see a couple of fish that are tucked into this coral. And a little close-up on one of these, okay, got to get it right, short raker. And a halibut swimming in here, settling down. Quite a lot of halibut up in this area, good fishing ground. So this study is working with DFO to create some rigorous techniques. The particular study, in a nutshell, showed that rockfish really like to be tucked into sponges and corals, whereas the thorny head, a related group, doesn't really care at all. They're scattered out on the seafloor and anchoring themselves on the seafloor with their pectoral fins. This is a program that's part of the Canadian Healthy Oceans Network that Dr. Driver mentioned, and Paul is the director of this network out of Memorial University. And so this is one of a whole bunch of different projects as part of CONI that are scattered across the country in all 3 oceans. And I'm just going to show you a little, well, all of my images are actually from the Pacific. I'm going to show you one little clip that was taken on a cruise that Paul was on this summer on the East Coast, where they were also looking at corals. But this clip is going to start off with the ROV descending down through a school of squid. And if you look just down at the bottom, we've got a swordfish who's going to come in to the image down at the bottom here. There he is, just in here, and he's focusing on this little squid here. He just gave it a good knock, and it's kind of stunned, and now he's knocking at it again, and you can see the squid ink. It's still moving, but now he's going to circle around as the rove comes down and deliver the coup de grace. Isn't that wonderful imagery? So now we're going to go on another CONI-sponsored project, but now we're going down to the Juan de Fuca Ridge, about 200 kilometers off the southern end of British Columbia. And here's the Juan de Fuca Ridge is a spreading ridge. It's a place where new Earth's crust is being created, and the Juan de Fuca plate is moving eastward and subducting under Vancouver Island, creating our earthquake country here. And with that magma rising up, very often there's magmatic eruptions offshore. For instance, this lava was created about 4 months before we got out there to photograph it. And associated with that heat source, we often find hydrothermal vents as well. And hot vents are formed as seawater leaks down into the crust. Is really leaky, so seawater gets underneath very, very easily. And as it goes down, loses its oxygen, it starts to dissolve metals and minerals from the surrounding rock into it. The hydrogen sulfide builds up. The water goes deeper, it gets hotter and hotter. And that buoyant, really hot water needs to get out. And if it can find a straight conduit, it comes directly out, blasting out as a jet and forming what's called black smokers. If the water is somewhat diluted by other circulation, it'll come wafting out as warmer water, replete with lots of hydrogen sulfide. And the chemistry of this water is such that there are bacteria that can use the chemical energy of chemical reactions and get that energy to fix carbon dioxide in a manner very similar to plants. So it's an analogy to photosynthesis. And that water coming out forms black smokers, such as here, where we've got chimneys forming, and that black smoke, when that hot water hits the surrounding water, which is about 2 degrees centigrade, here at 2,200 meters in depth, it instantly precipitates its load of minerals and metals, and it bonds to form little particulates of sulfide. So that's where the smoke forms, and it also makes these big chimneys. And here you see it's surrounded by animals who are feeding on the bacteria in the water. So I'm going to show you a little movie clip now taken from the Endeavour segment in the north end of Juan de Fuca Ridge. And this is the first time marine protected area was declared on a hydrothermal vent environment in the world, basically saying, hey, these environments really are special. So we're going to start out here with diving down a deep sea morid. And behind here we see basalts. Basalt is a very common deep water rock. These are pretty old basalts, and it's got a dusting of sediments on the surface. A deep sea skate here. Here's our laser 10 centimeter scale here. And now we're going to come into one of these chimneys. These chimneys can grow up 40 to 50 meters in height. In some countries of the world, the concentration of silver, metals, semi-precious metals in these deposits is high enough that those countries are thinking of mining, and New Guinea is actually already mining these deep sea deposits. So here we see a black smoker. In the Canadian deposits, by the way, it so happens that the geology is such that the concentrations are not that high, so it's not an issue here. Another chimney forming here with the black smokers just belting out of the top, and bacteria forming all the way along the sides, being blasted off here. Another smoker, and we see animals all the way up the side of this chimney feeding off the bacteria. So warm water coming up here, group of polychaetes. As I talk, watch these two guys here. They're fighting. The water is feeding bacteria that are growing all in this white stuff, and these worms are feeding on the bacteria like grass. And so these two guys are fighting it out here. You stay away from my grass, and I'll stay away from your grass. And so they're bacteriivores, and they're constantly grazing the surface. Now, these particular little polychaetes are among the world's highest tolerance of temperature. They're record holders. They can go up to 60 degrees centigrade. Here we are with a little vent at 300 degrees C, and these are the vent tube worms. And I'll just point you, first of all, to these little things that are flying in the breeze here. These are sperm packages. The males are sending out their sperm on long on long lines fishing for females. And you would never have seen this without the high definition. These are the gills of the tube worms, the red gills filled with hemoglobin, picking up oxygen, hydrogen sulfide, and carbon dioxide, taking it down into their bodies, and they have no guts, but they have a body full of bacteria, and those bacteria are undergoing chemosynthesis, creating organic food for these worms, and so that's the basis of these huge deposits, great biomass of tube worms going through this area. These little skinny ones are the same species, but they've been grazed off by mollusks and polychaetes, so they're not doing quite as well. So here we also are trying to collect another species which also has symbiotic bacteria in it. These clams also do the same thing with symbionts inside them. But unfortunately, we have somebody who's objecting to us collecting in his back garden. This is a deep-sea spider crab. This is a species that was only known from 2 broken specimens before we found them all over the place out on the Endeavour segment. And they're quite common, and they're quite aggressive. They will go after the vehicle. This is a female, and she has a little spider in her claw right here. She also has a little bit of an itchy armpit that she's just trying to deal with there. And then, sorry about this. Let me see if I can get this going. No, my pointer's not working all that well here. So here are 2 spiders. They're mating. The guy on top, there's 16 legs going on there. And she's just taking care of that. Now we've got a close-up on her face here with all her mouth parts showing and her antennae moving away here. So one of the things that we've been developing with the Rove pilots, and this is not oil industry class piloting. These guys are absolute artists when they're manipulating and trying to get us in working around these really difficult environments. And so over the years, we've developed the manipulative capabilities. In this image here, we're sampling in one of those really hot water vents and drawing water samples through and doing chemical analyses on the front of the vehicle. Okay, so now we're going to flip across the Pacific, and we're going to take our ROV and a ship, and we're going to go to the Mariana Volcanic Arc. And in this, we're sailing in right now into the sunken caldera of a volcano. And this is going to be using ROVs in a situation where you could never use manned submersible. It's pretty dicey in some of these areas because of the volcanic activity. So here we are on the Mariana Volcanic Arc is here. The Pacific Plate is moving westward. The Philippine Plate is moving eastward, and the Pacific Plate is diving underneath, forming the Mariana Trench. And as it goes down, it's melting, and that remelt is coming up, forming an island chain, forming a line of volcanoes, some of which form islands, but many of them are subsea volcanoes. And this has given us a wonderful opportunity to look at the interaction of the geology, the geophysics, microbiology, chemistry, and the animals that can cope in an actively volcanic and hydrothermally active environment. This work was sponsored by NOAA in the Ocean Exploration Program. And I'm going to today just talk about one particular volcano here, although we've worked along the entire arc. We're going to go to Northwest Rota Volcano. And it's so spectacular. We've been there now 5 times with a bunch of different ROVs. This is Northwest Rota Volcano. Down at 500 meters, it builds up to a summit at 550 meters below the surface. And when we were first out there with the Canadian sub in 2004, we found a pit at the top of the volcano, a volcanic pit. And from that pit, these huge billowing yellow clouds were coming out. And we couldn't see very well, so we thought, well, we'll just go in and find out what's in there. So we took ROPOs into the pit, and with the sonar, we could see it was 20 meters below us. But as we went down, the temperature was going up, and the pH went down below 2. So we realized we were in a sulfuric acid bath at that point and pulled the sub. When it got the submersible back on deck, you can see it's covered with this molten sulfur blobs that congealed on the sub. It took a long time to clean that off. So we went back, when we went back 2 years later, this whole side of the volcano had collapsed off, so we could see down into the pit and actually see the eruption itself. So here we are on the bottom, this time with Jason from Woods Hole, and now we can see the rock being thrown out onto the sea floor and the eruption itself happening. And so this imagery was filmed in 2006 from Jason. And at this time, we also put a hydrophone down on the seafloor and then synced the sound with the imagery. Now look right in the middle of the eruption, and you'll see flames coming out. Hydrophone's about 100 meters away. We're about 2 meters away. The yellow is sulfur, and the rocks that are flying out were just made. So they're coming out from the bottom, down deep underneath and coming out. So this is a really active eruption. If this was on land, we couldn't get anywhere near it. Because of the weight of the water, we can actually get in to see what's going on. There was at some part of the dives, the eruption was getting too violent, so we did have to pull out of the area. As a matter of fact, the first time we saw it, we were unsure whether the whole volcano was going up, so we pulled the ship out of the area entirely. Don't you love that just visceral pounding that's going on? I love that. So that is the first time anyone has ever seen an eruption up close. On Mount St. Helens, you just can't get in there, but it would have looked like that. So because of that, the National Science Foundation in the United States funded another cruise back there. So we went back in 2009, and this time took 2 HD cameras with us, and one is sitting here on Jason, and the other one we put both above Jason and then moved it and put it onto the arm of Jason as well. This camera was funded by CBC and formed the basis of one of the episodes of Nature of Things that was aired just recently. So here's the submersible going into the water. And as we go into this beautiful tropical water, crystal clear, nothing like Strait of Georgia. And here we are looking down on Jason. The submersible is here, going out into the darkness. And here we're looking at the arm of the submersible that's sampling in here, and the volcanic vent over this side. The vent wasn't quite as active last year, but still, throughout the time we were there, it would go through phases of activity and quiescence. So you'd think nothing would live in this environment. You turn the sub around, everything's coated with bacteria. There's hardly any species that live here, but the guys that are there are amazing. And this is a little shrimp that is only found on active volcanoes. And then this little crab who just leapt and grabbed a little baby of his own species. And I'm not going to show you the more gruesome part, but he tore it apart and ate it alive. Yeah, it's kind of sad. This particular shrimp is an amazing animal. We've now looked at volcanoes throughout the Western Pacific, and if those volcanoes are active, that shrimp is there. Now, in 2009, when we were there, the edge of the volcano was in through here. We went back in this spring, and all of this back part of the volcano had disappeared. There was a huge submarine slide that took all of that stuff away, and it collapsed away, all of the vents destroyed, most of the shrimp habitat gone. And yet, so this is the exposed wall, rock that was just been exposed. We go down to 600 meters, bacteria everywhere, and it looks like it's got the measles. And so you look closer, and you realize the whole surface is covered with these little tiny shrimp that have settled. And they're only about 1 centimeter long. And they're little baby shrimp that have just come en masse to settle into this environment. So the work that we're doing in my lab right now is actually working on the genetics of this species and trying to understand a little bit about what it is that makes this animal be able to cope in these kinds of environments. We did a whole lot more work on all these stars in through this area, and that work, plus some work by others on coral reefs in this area, contributed to a push to get this whole area designated as a national monument, which is what happened at the end of right at the end of Bush's administration. So that was actually quite a wonderful thing to have happen. Now and we transitioned from Bush into Obama, and Obama's administration was looking for a key to try and start a diplomatic mission with Indonesia, a very large Muslim country, and Obama himself had ties to Indonesia. And so this group that I was working with in the States, with NOAA in the ocean exploration program, they were asked to begin a mission that would use ocean exploration as part of that diplomatic mission. So they asked my group also to help them sort of plan this out. So with the Indonesians, we planned a cruise into the Celebes Sea. And for those of you who might know anything about diversity in the world, this is the center of biodiversity in the world. And especially in the shallow oceans, the numbers of species of fish and corals and sponges just beats anywhere else. But only once has any deep-sea expedition gone into this area, and that was in the 1930s, a German expedition that trawled through that area, bringing up broken, battered specimens from down deep. So NOAA sent its brand-new vessel called the Okeanos Explorer. And notice the big dome on top. It's a satellite transmission dome. And as the ship left from Bitung in Sulawesi, the governor brought out a group of people to bid the ship farewell, and they did this wonderful dance. This gentleman has a monkey skull around his neck. And I would like to say that it was the highlight of my life in terms of a place to go, but I didn't get to go. Instead, I was stuck in a dark room in Seattle. NOAA's new idea of ocean exploration is diving by telepresence. So there we have the submersible on the seafloor in Indonesia, sending the imagery back to the ship. The ship is telemetering it to a satellite, and it's coming back down to us in Seattle. So I didn't get to go, but nonetheless, it was still incredible. And so this was the INDEX-Expedition, and the submersible on board was this little Hercules. But this was the very first time it was done. So there was a lot of angst in D.C. and a lot of holding of hands to make sure that this was going to work. But all they told us was just for crying out loud, find neat stuff. Now that was actually fun, you know. And so they had 2 command centers, one in Seattle and the other in Jakarta, where the chief scientist was. He wasn't on board. But of course, the enterprising graduate students in labs that really wanted to participate very quickly, with the technicians, figured out how to hack into the satellite signal. So very soon we had the signal into our lab at UVic. I went back to UVic. There were only 2 scientists on board the ship, and they traded out, so it was only one at a time. And one of my grad students got to go. So there's John on board the ship, and he's directing the ROV pilots from the information from the scientists on shore. And he's waving to us, and here's Jonathan in my lab setting up our dive. We would dive from 6 in the evening to 2 in the morning, while Indonesia was diving during the day. So we got to participate. What lies below? I can't tell you how incredible the things were that we saw. It was just a fabulous trip for me, just seeing things that sometimes you see. I use the Challenger reports from the 1800s to do some of my work, and I saw things that I know have never been seen alive. So what I'm going to do here is show you a little video clip. And here we go with the little Hercules going down. Again, we've got a camera up above. Now, the Indonesians really wanted to find hot vents in their water. And so we had a good idea where to go the very first day. And indeed, we did find this cloudy water and came into these white smokers with lots and lots of molten sulfur pouring out in this area. And here's rivulets of molten sulfur that had just come out. And guess what? We got shrimp again, a different species in this case, but lots of bacterial mat with them grazing the mat on the surface. So hopefully next year we can collect some of these. But most of the expedition was spent exploring the deep areas of the ocean. And look at the tracks here going through. There's a little animal on the end of this. And it just amazed me just the degree to which seafloor processing happened. And the other feature was that every time there was something sticking up, there was some animal that was going to take advantage of it. So here's a coral with a little crab inside, getting up off the bottom, creating structure, and working with currents with turbulence around them. So here's a hard coral wrapped up, and all of them had their own ophiuroid or brittle star wrapped around them. I fell in love with crinoids, sea lilies, and feather stars. Stalked crinoids. Hardly anybody's seen stalked crinoids in the diversity that we saw them here. And this guy, this is a basket star, kind of similar to ours here, but quite a lot bigger. It's this one individual with all the little tendrils waving around trying to catch zooplankton. And a coral with animals on them. The glass sponges were also pretty amazing, things that the spongeologists have never seen. And usually when we would see a sponge, we would see feather stars with the sponge. There's 4 different feather stars on this particular sponge here. Oh, this guy. This is a carnivorous sponge, believe it or not. And it has pseudopods that come out like that are sticky, and they grab little organic particles, and they grab zooplankton as they go by, and they digest them. We call this a Sasquatch sponge. This is another glass sponge with feather stars on it, and each feather star has its own brittle star with it. The Dr. Seuss sponge, I don't know where that name came from, one of the ROV pilots. This is also a crinoid, like a pinwheel, and we're working with some crinoid specialists to try and, this guy, try and identify them. I got to stop this. Paul, do you recognize what it is? No, okay. This is a brand. Almost everything we saw is a new species. This is an enteropneust, otherwise known as a spoonworm, which doesn't help anybody either because almost no one's ever seen these guys. This guy's about 1 meter long, and you can see what he's doing. He's eating and pooping. The whole seafloor is just covered with the fecal pellets of this animal. And they're just little mud machines and just gorgeous animals. And we know this seafloor is just being processed over and over by animals like this. We saw 2 different species of these. This is a little hermit crab, and he's got a commensal zoanthid anemone on his back. Here's another one with a single anemone this time. I don't know what this is, but it's got a little cute thing hanging off its lip. This is a tonguefish, working with a guy in the Smithsonian, new species, chimera. It's a cartilaginous fish. Oh, and a gorgeous little squat lobster with the little furry arms, kind of related to the yeti crab, but it's orange. Right? Isn't that sweet? That's a male spider crab protecting the female who has eggs. He moved in when the sub came close. He moved in and grabbed her. And there's another female with eggs. And you can just see her antenna with a little hydroid attached to it up here. What we would do, this is a new species of squid. We would capture the imagery, and as soon as I would see them, I'd post it on SquidNet, and we were being followed by all these cephalopodologists who would then identify our squids for us, like this cuttlefish, and let us know if it was a new species or they would think it would be a new species. We couldn't collect any of these. And this is amazing. This is a sea spider, and he's about this big. He's about 15 centimeters across, 20 centimeters, carefully cleaning one of his arms. Little species interaction here. Oh, and isn't that lovely? A goosefish. So it's like a deep-sea anglerfish with this huge mouth and the gills that inflate to be able to drag in the prey. And this is another species. Look at the legs. It walks, and we would see it walking along the seafloor. Another modification of the pectoral fins. This is a tripod fish. Just gets them up off the bottom. Oh, and I forgot to look up what this guy is. It's pretty ugly. If anybody has any ideas, let me know. Oh, and watch this. Watch down at the end. Right. Sea cucumber felt us coming in the vibrations and up, off, swimming. And here's another one called a sea pig. It's also a sea cucumber. Gets down, takes a gulp of sediment, and off it goes. Isn't that just gorgeous? So, and I just go back to this. This is where I teach this stuff, but this is where you really understand ecosystem services. These animals are processing the seafloor, recycling nutrients, getting that carbon moving into a different form, getting it into fish, getting it back up into the water column. And this guy's a perfect example. He does a face plant down, takes a gulp of sediment, and you can see the mud all through its gut here, swims along, deposits the sediment out the other end, and off it goes. So we're really working on trying to get the Indonesian students involved, and we're hoping to go back again this summer and just to help them to discover their own deep ocean. Okay, so exploration does have other kinds of dimensions, and I'm going to bring back here to British Columbian waters. And there's other ways to probe the ocean other than using cameras and manipulators, and particularly wondering what's going on when we can't be there. So how can we put stuff out there so that we can get the information in real time back here and be able to interact with the ocean and be able to share it with people as well? And so this is one example of how to do that. And I work with this project called Venus, and my next-door neighbors are Neptune Canada. This concept of using telecom cables developed over a decade ago, and it just so happened the stars aligned such that Canada could do it first. But there's also projects in the United States, in Japan, in China, and in Europe that are following hot on our heels here. Well, some of the advantages here are that with a cable, you can send power out. You can put instruments out into the seafloor that you could never run on batteries. And not only that, you can bring back the information and be able to do it in high bandwidth rather than the lower bandwidth which we're usually stuck with. And we can begin to share things in a much better way. And so a student in Halifax was using our camera in Saanich Inlet on the island. The camera's down at 100 meters in depth, and she's moving that camera around as part of her project to take photographs back in Dalhousie. So Neptune Canada is an example of the far end here, and their vision was to instrument an entire tectonic plate. So here's going out to the Endeavour segment that I've talked about already. And they have now deployed cables in last year and this year, instruments at 5 locations down into the deep basin and then up onto the shelf. And they've done a huge amount of stuff, but I'm just going to give you one little example here. So here's their big node going into the sea, into the ocean, with the backbone cable connected to it. The submersible then comes along and plugs in the instrument platforms. And so here's the cable coming back to the instrument platform, and on that platform we see a bunch of different instruments such as the sonar, such as this current meter here, plankton pump. And so those are all wired in, wired back to shore, and wired to a data repository and to the scientists that are doing the research. one small example. Here's Ropos going into the water, and slung underneath Ropos is a little crawler that Ropos put out on the seafloor, connected into Neptune, and it got the name Wally. So Wally the robot here is going across the seafloor, and it's being driven from Germany. And on the front of Wally is a camera and a bunch of sensors that can measure oxygen, methane, temperature. And so here's an example of the data from Wally, where we've got, Wally was moving here. We got 4 weeks of data going across the bottom. Wally was moving around a little bit, but this up and down of the pressure, it's actually the tides going up and down on the surface, so you see our semi-diurnal tides. And we see temperature changes also, partly in response to those tides. The methane sensor, however, was flat, and then all of a sudden saw this spike in methane. So here's a little video clip actually taken from Ropos, not the Neptune cameras, and I'm going to just whiz it along here. Here you can see the bubbles coming out of the bottom. And we've got a little fish coming in here. He's going to investigate the bubbles. It's a zooarcid eelpout and doesn't like those at all. Those bubbles are methane. Underneath the sediment surface is methane clathrates. It's ice water that's bound methane in an ice form. And it's subliming and coming out as gas being released up through the water column. So there's the little bubbles going up to the surface. Okay, so let's come right back home now. And here we are back on our friendly Strait of Georgia with its muddy Fraser River plume coming out. Is there much left to know in this area? So this is the homeland of Venus, where we've also deployed some cables, much shorter ones, and we have a cable going out from the Iona sewage treatment plant, a delightful place to have a shore station, out into the middle of the Strait of Georgia, where we've got a number of sites under the plume, and we also have a little array here in Saanich Inlet as well. The researchers that designed Venus really were interested in knowing how our local ocean worked in an area where there's a lot of human impact. And so that was the design with the instrumentation. We're currently also instrumenting out, putting instruments on the ferries so we can know more about the surface. And we'll also be putting in coastal radar on the end of the coal port and a bunch of other gliders and AUVs associated with it. So anybody can get the data off Venus. It's open and free access data. This is one of our sponsors for this evening, so I thought I'd make sure I put it in. We've been working with local industry to help develop the capabilities. So they built our nodes, our big switching boxes on the seafloor, and OceanWorks now has an entire arm of its company dedicated to marketing these, and they've been able to make a number of sales around the world for deep water communication hubs. And so we're the same kind of idea as Neptune. You've got an array of instruments with data coming into the node, and from there it's being sent back via high-speed internet to a repository at University of Victoria and then out to the scientists. And here Rachel is looking at data and a camera feed from Saanich Inlet. So we'll just take a closer look at what she's doing. And so this is a pretty amazing story if you think about the migration that zooplankton do. And every day, zooplankton in our water swim from about 100 meters depth, and at nighttime, when the sun sets, they go up to the surface to feed. So that's 100 meters of swimming, and you're only, a few millimeters long. So that's a lot of body lengths. I pulled this image off just this morning. This is what happened yesterday in Saanich Inlet. So this is the bottom. It's an instrument on the bottom looking upward to the surface, like an echo sounder on a boat. So here's the surface of the water, the red line. And we can see it at sunset, the day before last, the plankton went up to the surface, fed during the day, and then came back down as soon as the sun came up and spent the day at the bottom. As a matter of fact, I think some of this red stuff is fish that are feeding on the plankton because many of them didn't go up to the surface. And there are a lot of zooplankton in Saanich Inlet. This is a very, very rich place for zooplankton. Most of these in this particular image are krill, the Euphausia pacifica, and you can see some herring swimming by here as well. And so as we look at zooplankton patterns throughout the year, these are images from different days, and you can see that there are times when zooplankton isn't very abundant. We've probably got something weird coming in here like amphipods. And then here we've got the big spring bloom happening, and we can see phytoplankton just raining down. And then as summer is setting in, less phytoplankton, and you can see the night length getting smaller, so the zooplankton are up at the surface shorter length of time. And so you can begin to study the dynamics of the migration and density of the zooplankton themselves. And these are the guys that are important for our salmon. This is what the salmon juveniles need to feed on when they emerge from the streams. One of the really important things about these observatories is gathering baseline data. We know our oceans are changing. We know they're warming. They're acidifying. This is 4 years of data from Saanich Inlet from just 4 sensors as we're looking at changing throughout the year. So you can see temperature going up and down with seasonal change. And look at the oxygen. Every single year, the Strait of Georgia is renewed as upwelling water off the coast brings nutrient-rich and oxygen-rich water into the bottom of the strait. And that happens in the fall. And as that water moves in, it rejuvenates all of the inlets around, including Saanich Inlet. And so we see in Saanich Inlet the oxygen falls to almost zero during the summertime, and then it waits to be reinvigorated with oxygen as the strait fills up as well. Something weird happened last year. You can see this in the temperature. It didn't cool down, and the oxygen didn't really get up there. So that was a little bit worrisome because now we're not getting the inlet totally renewed. And so we're watching now to see what's happening through the wintertime here. But these kind of data are what is going to be needed over the next decade to really have a good look at what's happening. And we're gathering the same kind of data from the Strait of Georgia. Strait of Georgia is a busy place, lots and lots of shipping going up and down in this waterway. And one of the things we're doing here, working with some whale researchers, is putting hydrophones out in the bottom of the inlet to listen to what's going on. Whales out in this area clearly have a little bit of competition for the acoustic environment. So I'm going to play just a little clip here, and the first part of the clip is Saanich Inlet. And then the second part of the clip is Strait of Georgia set at exactly the same gain. So you have to listen hard to start with. Bit of a difference, isn't there? So the quiet ocean, you can hardly hear anything. You could just hear a little crab. And the sound that you heard is this stuff. So this is a sonogram. This is what sound looks like, the shipping. So really broad frequency sound in here. So what happens when our whales go by? This is a few seconds of whale chirping. And it's embedded, and this is quite a few, let's see, what are we looking at here? Oh, we're looking at 300 seconds here. So you see the yellow is going to be masked if you think about sound here. And I'll just play you a little clip here. That's the ship, is actually out in quite a way away. And the whales are closer to our hydrophone here. So one of the studies that's just beginning is just addressing what these issues of, is acoustic pollution something we should be caring about? So as I just come to an end here, our goal with Venus is to try and bring that, okay, you can come from anywhere to do research in a waterway that is really important to us, but also really used in many different ways. And just to end off here, I'm going to show you a little bit of a clip. This is not HD imagery. I let one of our technicians play with it with $8,000 to put a webcam down just to see whether we could use it. And Gail Anderson here from sorry, I said the bad word, didn't I? Bite my tongue. From Simon Fraser is working with us because she's doing some work in forensic research to look at what happens to bodies that are disposed of in the ocean. And she uses pigs as her proxy. So you're going to see a dead pig here. And I want you to also remember this is Shark Week, right, in the newspaper. So I'm going to show you a little clip here. Gail did not even get to see this. It happened so fast. So that is a large 6-gill that came in, and this is approximately 2 hours after we deployed this pig. We're still trying to install the structure, and my project manager's going nuts because I'm yelling at him to take these pictures, and we're still trying to plug things in, and the sharks have already gotten the pig. Meanwhile, Gail's at a conference. She wasn't going to log in until Monday, and this is Saturday. They've already gutted the pig here. The pig's been tied down so that it doesn't get taken away too easily. This is actually a fairly small, and these are 6 gills. This is a fairly small shark. It's only a juvenile, probably about 1 meter long underneath the camera. And it's just sniffing around here. The little buzzies that are going around are amphipods. This is the neck of the pig, and it's trying out the nose, just giving that a little shake here. No, don't like the nose. Let's go into the neck and just nosing under the skin, just trying to get a good bite. Okay, now we can get a good bite here. And look how they work. So these are scavenger sharks. They're not predatory sharks. They don't have great big teeth that come in and take a wonking bite out. They're ripping the flesh. Look how it's going at it, turning and ripping to try and get the flesh off. And so that's the hallmark of a scavenger. These sharks do a really important service. I mean, they're cleaning up on the seafloor and, again, recycling carbon. And he's just determined to get that good bite in there, and so he's just got a grip and just it's interesting to look at how the body is turning and going back and forth as now he's getting it. Now he's getting that piece off. And you can see the 6 gills as I'm going to get this bite. It's the last thing I do. Come on, guy. You can do it. Yes. Come on, it's getting late. He's got it. But he just swims forward and he gets his lower jaw caught under the little rope there. So he's having a little hissy fit here. And then another one comes in, and we've also had a big shark come in underneath. And this guy's really going at it. Now he's vertical and he's just corkscrewing, trying to get a piece out of the neck, and just the fur is flying here. So it's pretty amazing to see how, look at, he's just vertical, just trying to get that out of there. It's pretty amazing to see how they work. And there goes another big shark by the side here. The pig lasted, we had to move, we had to shut it down, and we got back on 15 hours later, and it was gone. So that was the end of Gail's pig. Gotta get it. Yeah, I just have to let him get this last bite. There's that guy going by there. Sorry, I love it. Okay. That's that. All right. So just to end off then, just the message here is that there is a whole lot we have left to learn right around here. And part of that messaging is there's a lot going wrong with our oceans, but if we can learn to appreciate it more, understand what's going on, maybe that's a major step towards making better decisions. And we've got a lot we can build on with the technology that we can contribute. So with that, I would like to thank Pat in particular. Thank you for having me. And also a thanks to our sponsors and supporters. And thank you very much for coming.
Well, let me just say that was just a great lecture. I could sit here for hours and watch those videos. We do have a couple of people who we've asked to respond tonight, and so perhaps we'll start with John, and if you'd like to give us a few comments. So over to you, John.
Thanks, Verena. It was the images are amazing. I want to focus just a little bit on the fact that this, that the province has declared this year of science. And science is an interesting word because there isn't just one science. There are all kinds of sciences. And I don't think we can have a presentation that would illustrate more of those kinds of sciences in one place than the presentation Verena just gave us. Because clearly it's not just some of the formal sciences of biology or ecology. There's the whole set of sciences that ranges from engineering to hydraulics to telemetry in the subs and the ROVs. Of course, the ships that take them out there are packages of engineering and science as well. And then the activity of exploring is a science-based activity. And so I think in the work that's been done around Year of Science, some of the polling data shows that people have a conception of science as a laboratory science, as a laboratory activity. Well, Verena's just proved that it's about as far from the test tube and white lab coat as you can get, and that people aren't sure what kind of a process science is. And of course, many people in school were taught that science is testing a hypothesis or a theory. And yet, clearly there's a great deal we don't know, and we need to go exploring to do that. So we, in our organization, we occasionally have a discussion about, well, is exploration science or not? Clearly it is. You don't know what you're going to find, so it's hard to test a hypothesis. I guess if you're testing a hypothesis, it's if we don't go look, we're not going to find anything for sure. And if we do go look, we don't know what we're going to find. And so I think it's very exciting that, well, I think it's great that we have Verena in BC and that we have the undersea explorations. And I think in terms of students in BC that there's an incredible range of future careers across more kinds, across any number of disciplines and more kinds of sciences involved in them than any of us would guess at first. And I guess I'd like to just finish off by saying that I think that these kinds of this kind of work, this kind of exploration, research that brings back not only a better understanding of the environment in the sense of new species, our understanding of diversity and abundance in the world, but the compelling images help engage people who aren't used to trying to look through that water surface, and as a result of not trying to do that, really don't care much about the ocean. And so I think the compelling images and the stories that go with them, and I've heard Verena speak before, and I know one class of students that I heard questioning her about how she got started and how'd you get into this and how do you do it and how do I get started, is a kind of a whole other set of subjects. But, as we were talking beforehand, Canada has the longest coastline in the world of any country. We've got an amazing amount of ocean and coastline. We don't have very many people strewn out in a great big country, but we do seem to have a pretty remarkable amount of ocean sciences underway, and I think we all want to keep it that way.
Thanks, John. And Paul, your comments maybe from an East Coast perspective.
Well, thank you, and thank you, Verena. What a voyage. Wow. I heard lots of oohs and ahs from the audience and found, in fact, that I was making some of the oohs and ahs as I saw some of that imagery, even though I, too, work in some of these sorts of environments. So it's just spectacular. I think what we've seen tonight is that Verena's talk really illustrates that the age of discovery for the oceans is happening right now. We saw tonight all sorts of new habitats, new species. Some of these things have never been seen by anybody before these videos were collected. And also new processes, the ways that these species relate to one another and influence each other. And so in remote areas of the world, it's very easy to find these new species. It's a lot of work, but nonetheless, many of the samples that we bring up have these new species. But even in relatively well-known waters, like British Columbia, that potential is still there. And there's still tremendous things to learn about the activities in the oceans. So I think all of us, I would hope, realize there's a lot to be excited about in terms of the oceans. But at the same time, there's a lot to be worried about. So we see spectacular imagery of coral, for example, just north of here. But then we also hear that fishing is having a negative impact on these corals, and that we need to be more cognizant of what these corals do and what we can do to preserve them. Elsewhere, we've heard lots of bad news about the oceans. We've heard 90% declines of top predators in the open ocean. We've heard of collapse of global fisheries by 2048. And we've heard that there could be loss of many reefs around the world within 50 years. And then I'll also mention ocean acidification and warming, because these are concerns that will actually impact things in the deep ocean, including some of these spectacular corals that Verena has just shown us. The good news, though, is the explosion of technologies, many of which were illustrated tonight during Verena's talk. And I would say that almost everything we saw in tonight's talk really wasn't possible even a decade ago, or at least not to the level that it was tonight. And so I think we really need to appreciate how far we've advanced in ocean studies. So today we have cameras that use sound and require no light in order to collect images. We have robots that we can program to run missions and then bring the data back to us on shore. An example that Verena gave us is underwater vehicles that send high-definition video back to shore via satellite. And so I, for example, can sit in my study back in Newfoundland and see the seafloor off the coast of British Columbia in real time while I have a dog curled up at my feet. And that really is a fundamental change in the way that we do science, which I think is really spectacular. We've also seen glimpses into very distant worlds, and I'd like to use that particular observation to point out the importance of international collaboration. And so the example that Verena raised was Indonesia and the United States, but the key point here is that through this collaboration, the scientists of Indonesia were provided with access to really cutting-edge technologies that are really, really brand new, and are certainly technologies that they would not have access to otherwise. At the same time, we, as scientists in the West, really want to access some of these environments that are hotspots for biodiversity that are often very far from our coastal waters. And so in order to do that, we need to create these collaborations and try and break down some of the protectionism attitudes that are starting to develop around the world, where there's fear that perhaps scientists are trying to go in and steal biodiversity or the resources associated with that biodiversity. And that is something that needs to be considered, but at the same time, it also impedes discovery. And people like Verena want to go in there and just see interesting, exciting stuff, exactly as she said. And so allowing that to happen, I think, is very important. Now, the audience that's here tonight obviously has an interest in the oceans, and that's just fantastic. And keep coming to these sorts of presentations, because we need to do a lot more for the oceans. There are people like Pat Gallagher and Gordon Slade who've worked year after year to keep this series going. There are people like Verena who have done so much to enlighten us about life in the oceans, and I mean me as a scientist, but also you as a general public. And we can see her love for pooping Ontrop-nest worms and hungry sharks. It just comes out so clearly in everything that she says. This is really cool stuff, and as scientists, we're really privileged to be able to do it and to be able to go out there and do this sort of research. But we need to convince you that this is exciting and important, and I don't think for a moment any of you don't realize the excitement in what we've seen here tonight. But I do want to reemphasize the importance of it. So Verena mentioned ocean health and the fact that many of these species provide key services that we really don't understand very well. And the example, for example, of the euphausiids, which are such a key food resource for salmon, this is a species otherwise people really wouldn't think that much about. It's just a little crustacean. It's not that attractive, and yet it's such a key resource for a species that we do care about. There's also a lot of untapped potential, and one of the examples that we often use in biology is tag polymerase, which is used for DNA amplification. And so this is something that was discovered from an extreme environment, not hydrothermal vents, but a sort of sister environment, that's now used in everything from crime scene investigation to paternity lawsuits and a whole variety of other things that have major impacts on our day-to-day lives. And so right now in Canada and elsewhere around the world, there's a lot of increasing pressure for deliverables. And so if we can't argue that this video is going to help us within 2 or 3 years, it's very hard to get the funding to go out and collect it. But in fact, this sort of video is very important, and its value is very hard to evaluate in the short term. Now, to have this lecture in Vancouver, I think, is extremely important. If we look at Canada, we're rather unique as a nation because most of our population is stacked up along the US border. And so most Canadians don't naturally think about the oceans. People in Newfoundland, people in PEI, people in Nova Scotia, they do think about the oceans, but people in central Canada don't naturally. The problem is that Atlantic Canada has a very small population. We don't have a lot of clout. But British Columbia, which does care about the oceans, does have clout. And so what I want to ask and finish up on is asking you to write to your newspapers, to your members of parliament, to your cousins and your friends who live in Toronto, and tell them that you care about the oceans and that the ocean's important. We can do better, and we must, because the oceans are surely worth it. So thank you, Verena, and thank you all for coming.
Thank you, Paul, and thank you, John, for your comments. We have some time for questions, and I'll take questions if people would like to raise their hand. We usually have a couple of rules here, which is that try to keep the question a question rather than another lecture. And if you could be fairly short and focused, that will give more than 1 or 2 people a chance to ask either Verena or one of our respondents to the lecture their opinion on various issues. So who'd like to go first? Yes, go ahead.
Thank you. What do you see as the biggest impediments to having some more success in this field? Verena, sorry.
I think it's directed at you. Do you want to come up to the main microphone?
Who asked the question? Sorry. What do I think are the main impediments for?
To have more success in developing this technology in this field.
The main impediments. Wow, I got to get the money answer out of my head because I'm dealing with budgets this week. I think it's making really, really sure that we have the connectivity and we have the support that and the engagement. So when people turn to you and say, "Whoa, that costs a lot. Why would we give money in that area?" So money is important, but behind it is the appreciation as to why we would perhaps do funding into the oceans rather than, there's only so much that goes around. So rather than perhaps some other area. So or can you inform some other area and be able to do some of those linkages? For example, art. We're actually working with some artists who are saying, "Oh, we're not getting funding." Okay, well, we're getting some funding, so we can help you. So what do I think is, it's really, that's one aspect, but one of the things that we've really enjoyed as scientists are really good at having interesting visions as to what they'd like to do, getting the engineers to respond. And that's often what takes a while. As Paul mentioned, this technology wouldn't have been possible. And it wouldn't have been possible without the scientists pushing and pushing and pushing. So that's one of the impediments, is to make sure the engineering can keep up with you. And so having the kids interested in going into those kinds of areas to be able to work with them in the future.
Yes.
To talk about one of the negative things for just a second, a few months ago, I was, and I'm sure everyone else here, was horrified, truly horrified by what was happening in the Gulf of Mexico. And yet I look at the hydrothermal vents that have been going on for millions of years, spewing all sorts of noxious things into the ocean. And I thought maybe someone from your perspective, seeing these things closer, you might have, you know, that little bit of oil spilling into the ocean might not have been such a frightening prospect, and you didn't have the sense that all the rest of us did, that the oceans were going to be dead in a few months because of some leak in the Gulf of Mexico. Okay, I wonder if you could address that.
That's a very astute question, actually. I started by saying we're visual animals. So the thing that horrified us so much was that we could see it. And it was on the surface, and it was oiling the birds, and we could see what that was doing. To be honest, my reaction was, you know what, that's an organic phenomenon. That oil is organic. And yes, there were some horrible things. And the worst part of it was the people's lives that were affected and the animals on the shore. I mean, that's the worst part. And I don't mean to say anything negative about that. But as a phenomenon of pollution, it's an organic problem, and nature has been taking care of oil spills for a long time. So eventually, that oil is going to break down. It's a question of how long and how much damage and what the economic impacts. That's another issue. What I think is far more frightening than any oil spill is things like ocean acidification. That is something you cannot sell in Ottawa right now because nobody can see it. We don't understand it. The chemistry's difficult. It's really hard to see the connections. And so from my point of view, I kind of feel like I do work in an extreme environment, so I see those really extreme things, but the question is, what can be remediated, and what is the length of time? So in a way, I mean, as I say, your question's astute because you step back and the oil spill will eventually go away, but things like acidification will not. They're already the momentum in the system, and it doesn't matter if we start now, we're going to see the effects of it. So a quick, perhaps trite answer, but...
There's another area of extreme environments, and that is methane hydrate, and this peculiar frozen form of an inflammable gas, I guess, has been around a long time, and it received a lot of publicity a few years ago, but we haven't heard for a while what that's about. I think someone wanted to actually harvest this stuff at one point, and if you could help us on that is, for instance, what temperature does it form at? Somewhere around 4 Celsius, around there?
That's right, yeah. So the methane hydrates that are found off the coast, they're on the shelf at British Columbia. They're somewhere around 800 to 1,200 meters in depth, and it's just that interaction of water pressure and temperature, and we've got all this organic stuff inside the sediments, and it's just sort of the perfect conditions to form methane hydrates. Possibly it's going to be a whole lot cheaper if one really wanted natural gas to get it from Arctic, because it's under the permafrost, a lot easier to get. There's a lot of concern about methane hydrates as well because it's a very strong greenhouse gas. And so one of the concerns about deep water warming is that a lot of those methane hydrates are going to be melted, and then all of a sudden a large amount of methane hydrates released. At the moment, there's not a lot of interest in trying to exploit them in deep water simply because of the cost of trying to get them out. But there are people thinking about the shallow water ones, particularly in the Arctic. But there you just have to think about what are you doing? What are you doing in terms of input in the atmosphere, and what are you getting out in the long run? What is the cost in the big picture? Did that answer your question? Because there is quite a lot of research still going on it. Yeah.
Have you seen any of this stuff in your submersible studies?
Okay, so the bubbles that I showed, that's from methane hydrates underneath. That little bubble clip, that ice is underneath, and I do have images of ice, and it's pretty weird to come across it on the bottom. You know, you're going along, there's an iceberg, it's sitting on the bottom of the ocean, and it really does look like ice. And so it every now and then it breaks off. If it breaks off, it floats up to the surface, and so you see these little ice flows on the surface. And yes, you can light it with a match. Yeah, it's pretty amazing.
Hi. Yeah, I just have a question in regard to, you were commenting on a little warmer ocean in Georgia Strait, and also the lack of oxygen the flushing happening this year. And we live near the ocean. Two things we noticed this year, it was reported in the news, these white-sided dolphins that are never up here were frolicking around in our waters. And the other one was that we noticed a huge algae bloom this year all over the surface, like a massive amount, never seen so much. And I just wondered if that was tied into warmer water, lack of oxygen in the water, that sort of thing.
Okay, it wouldn't be the lack of oxygen because the lack of oxygen is way down deep.
Okay.
So it's the deep water with the oxygen story. John, do you have an answer? Because you might be better at that than me.
Well, I mean, white-sided dolphins have been coming in and out of BC probably for the last 2 decades in bigger numbers.
Yeah.
They were historically, a lot of them in BC back at the turn of the last century, reported in the fisheries literature, and then they kind of went away and went offshore and came back starting with the El Nino, La Ninas in the mid-90s in bigger numbers. And they do seem to come and go, and they're quite visible. You see a school of 1,000 of them.
Yeah.
So that means that the food is there, or they wouldn't be there. So the fish that they're eating, the herring and whatnot, populations are up as well.
Okay, thanks.
Yes, at the back.
Turn that on. One of the things I worry about is the possibility of a series of positive feedback loops sort of taking off. Because I've seen examples of, for example, the more the ocean acidifies, the more greenhouse gases it emits. The more it acidifies, the more greenhouse gas it emits. And there's just examples of that throughout. Is there anybody studying the potential for these positive feedback loops to take over and just essentially collapse whole systems? Does that make sense?
I'm just working on the concept. Do you mean more acidification is putting more CO2 back into the atmosphere? Is that what you're...
Yeah, because I know that there's stores of methane, for example, that would potentially be released.
Okay, no. Okay, my understanding, the acidification is actually the CO2. So methane is CH4.
Yeah.
So we got methane here, and we have CO2 coming in. The CO2 is going into the carbonate system. That's the problem with the CO2 system, is that carbonate chemistry is a real mind-boggler. But what's happening is more CO2 in means you're shifting the carbonate system away from the dissolved form of carbonate, which is what many animals need to make their skeletons. That's a lot of this. That's the scary part there. I don't know how that chemistry fits into going down into methane, which is CH4. Methane, so...
Well, anyway, but maybe I can talk to you about this more
yeah,
after.
sure. You might be able to get
Because
me into
I don't think I'm asking the question quite right at the moment.
It's a good question. Who's looking at this? You know, this whole acidification thing took the science community completely by surprise. So it's been one of those things that we're scrambling to try and understand what's coming on, because the hypothesis was it doesn't matter because the ocean can buffer any CO2 we put into it. What we just didn't take into account was the rate at which the ocean needs to be able to do that buffering, and the CO2 is going in so fast that the ocean is not being able to respond. And so the people working on it, they're only just beginning to try and get into the system and understand it, and there aren't enough people working on it yet. So it's a good...
Yes.
First of all, I thought the footage was absolutely stunning. I just love seeing stuff that is brand new. I'm just wondering about the future of this footage. How will it be distributed to the inland public?
So the NOAA footage, NOAA is required to make anything available, and you can get anything you want free of charge from them. A lot of the imagery and the clips are available. The still imagery is available. As a matter of fact, the Indonesia trip, I don't know where that is at the moment because they wanted to make sure the Indonesians got it first. But the NOAA government, anything you want, it's available free of charge. Anything
is this on their website?
There are clips on their website at the moment. So, for instance, the expedition to ocean. If you go into Google on ocean exploration, you'll get to the NOAA website, and they have tons and tons of gorgeous imagery, still imagery and footage. And they've been trying to push it out to the public as much as they can. They're doing a wonderful job there. Anything off the... We're trying with Venus and Neptune also to try and get our imagery out. Boy, is it ever tough to do with video, though. It's so high, especially the high-resolution stuff. So that's problematic. Woods Hole imagery, you got to buy it. They charge a lot of money. And the CBC stuff is available on the CBC website. You can actually download that imagery as well. So, and then some of the ROPO stuff, it's basically we have stuff and we do give it out when we can. It just costs a lot of money to get it out to people, and it turns out it's not the right format, but it is available. And I don't know about Coney. We got to think about that, Paul. What are we going to do with the imagery? What is our data policy with respect to that? It's something we need to get to one of the committees.
I just have one final comment about that. You had a quote from Cousteau right at the very end that said, "People protect what they love." I think, you're an amazing scientist, but I think you're also an amazing presenter, and you should go into TV or something, try a new, the ocean nature of things or something like that. Just the way you talk about things is wonderful. Thank you.
I think that was a great comment to end on. And so just let me say thank you, first of all, to the organizers and sponsors of tonight's event. Secondly, to our commentators at the end of the lecture, but particularly to Verena for just a really wonderful and stimulating talk tonight. Thank you very much.